Okay, so it looks like we're just waiting for a few more. Right, okay, so can you hear me okay online?
Yeah.
Thank you. So in the last lecture, we talked about different scaffolds and different ways that we can form scaffolds, especially towards mimicking that ECM. So we looked at different ways of seeding those scaffolds, whether it's in vivo or in vitro. And then we looked at this electrospinning technique in order to form the scaffold. So we kind of talked to this at the end of the lecture. So there's also other ways that we can form these porous scaffolds. Another way is to use a thing.
Yeah, the screen is not sharing.
Sorry. It's all right. We've got 17 days left. I am going to get close to an immaculate lecture. Yeah, there is officially 17 days left, I think, of the whole semester. There's not 17 lecture days of this, so just to let you know. We're going to do this 3 times a week now. Okay, so there's other ways of producing these. You can see the screen now online, right?
Yes, thank you.
Okay, perfect. So there's other ways of generating these porous scaffolds, and the second way is to do this particulate leaching. So the particulate leaching method is to mix a polymer solution or the macrama in the presence of a salt solution. You can cure and cross-link the polymer. And then what you do is you evaporate the solvent to leave the polymer scaffold with salt crystals inside. And then you wash the scaffold with water in order to dissolve the salt and leave behind the pores. So obviously, these porous materials, we can modulate the size of the pores depending on what salt we use for these things. We can change the pore size, and we can change the shape of those things as well. So the great thing about these is that obviously, as I said, they're tunable. So there's lots of different variables we can tune. We know that if we tune the cross-linkers, we can tune the density of things. We can also obviously use those different porogens, which is referring to the salt itself, in order to manipulate the size and shape. These things are reproducible, and they're reproducible because of the size of the salt is consistent. And then you get controlled porosity. So you can have up to around about 93% porosity in these things. The pores are interconnected, and you can use those for the encapsulation of cells. Now, one of the issues is that you can have stress concentrators. So stress concentrators are these irregular shaped pores, and they result in stress concentration decreased mechanical properties. And that's due to the lack of interconnectivity between pores. So as you can imagine, not all the pores are created the same way in space. So it actually can give rise to weaknesses in the mechanical properties of that scaffold. And then the other thing is that if you don't obviously tailor the salt properly, you can have really large pore sizes in there, which obviously affects the flow of things in and out of those scaffolds. Sorry, hopefully you've got everything there. So another way to do it is a gas foaming method. So you can cross-link a polymer in the presence of a blowing agent. So chemical blowing agents yield gaseous decomposition products during reactions. So when we mix an isothiocyanate with water, we yield a carbon dioxide byproduct. That carbon dioxide, obviously, as it's released, will allow for the expansion of these polymers and the creation of the pores. We can actually also add a physical blowing agent. So we can add a gas like carbon dioxide. In this form, it's supercritical. So it would be a solution, a liquid of carbon dioxide. As that gas expands, it forms bubbles within that scaffold, and it does that during polymerization, which again leads to the creation of these pores. Now, the good thing about using this gas foaming method is, again, that they're tunable, and we know that we can change the cross-linking parameters. We can change the blowing agent, and we can also change the surfactants that we use in there as well. They help us modulate the pore size. The pores are interconnected. We can have a wide range of porosity, so anything from about 20 to 90%. And the pore sizes are wide ranging as well. So I don't know why it's written there 10 seconds to 100 to 1,000 seconds. So I get rid of the S, but it's 10 to 1,000 microns. So we have quite a big, broad range of porosities in there. So one of the issues is this, and you can see this on this bottom image here, A, is that you get this skim formation on the outside edge. And that skim formation can obviously stop on the extremes the flow of different agents in and out of those pores. Reproducibility is difficult, as you can imagine. Expanding a gas, it's really difficult to control where that gas goes. And generally for those things, you can't use them to encapsulate cells, but that's just the general terminology there. Now, one which has seen a lot of interest over this, over the past few years, is the use of 3D printing. 3D printing technologies have really come on, and you know now that you can buy a low-cost 3D printer for a couple $100, and you can basically have free range over what you're producing. So I'm sure some of you will use these kind of technologies in your senior design classes and things like that in order to produce different materials. So the 3D printing builds these 3D shapes in a layer by layer fashion to obtain complex geometries. There's lots of different types of 3D printing. You can have the filament based 3D printing, or you can have stereolithography, which generally works off resin. The great thing about this is you can make complex patient specific shapes. So that actually means that it starts to go into this area of personalized medicine, where, if there's a specific shape that needs to be developed for a patient, we can easily take images of that and transport it into something like SolidWorks or other tools like that, and we can then manipulate it so we can 3D print. We can design it so we can encapsulate cells, so we can have that porosity designed in. And we can use it with a range of different polymer types. So the common ones that you come across are things such as PLA and PETG, but actually you can also use flexible polymers as well for these things as well. Yeah. Now the problem is with this is that it can be quite a costly process, and it's costly in the fact that when you're trying to make a specific shape for a patient, it's not costly in just the fact that it's printing, but it takes a lot of time to get to that phase as well. So cost and time are two things that go together. Smaller scales are difficult, and I would say that that's in between now. So with a stereolithography type printer, you can probably get down to about 25 microns in size, so fairly small. But with a filament-based printer, you're probably in the hundreds of microns range there. And you can use those, you can actually use 3D printing to develop molds, so you can actually then scale things up a little bit easier. So there's lots of ways that you can use 3D printing, and obviously one of the ways that we can tap into that market is to print gels which support cellular matrices. So one of the ways, three goals associated with 3D printing and tissue engineering are to help direct stem cell differentiation, and the second is to recruit native cells, and the third is to direct tissue growth. Now, it's important in all them processes are cell proliferation and the migration and the differentiation, and they're influenced by 5 factors here. So they're influenced by the presence of soluble factors, such as growth factors, so things such as VEGF, which is a vascular endothelial growth factors, and PDGF, which is a platelet-derived growth factors in there. They can differentiate, they can control those cell proliferation, migration, differentiation. Then there's also the aspects of the cellular-cellular interactions and the cellular ECM interactions in there. The substrate modulus, obviously we know that now, right? We've been through this many times, that we want to try and match it as closely as possible to the material we're trying to mimic or replace. The mechanical stimulus, what kind of stresses and strains those things are going to be under, and also the environmental cues. So things such as hypoxia, where there is limited oxygen saturation in the surrounding tissues. Now, there's lots of different types of molecules which influence those three factors: the proliferation, migration, and differentiation. So we can look at small molecules, things such as corticosteroids. Corticosteroids have an influence over cellular adhesion. Things such as hormones, again, that kind of buys into kind of the growth factor of these things. We can have proteins and peptides, and that's the presence of things such as these cytokines, these growth factors, and the ECM molecules. And then we can have oligonucleotides too, so the presence of DNA, RNA, and also kind of the smaller substituents of those things. You might have heard of things such as microRNA and silencing RNA, and they're pretty important in some of the other therapeutic uses, and just nucleic acids in general. So all those things can have an influence over the three factors: that proliferation, migration, and differentiation in there. Now, here's one example of how we can temporarily control the release of different growth factors. So we have VEGF and PDGF. You'll notice with the vascular endothelial growth factor that we just mix it with the polymer, and we generate this porous matrix, and the actual VEGF just is suspended in that porous matrix. It's suspended around the porous matrix in the polymer itself. There's another way that we can do it, and we can actually incorporate the PDGF directly inside the polymer, and then we can make this porous matrix. And by doing that, we can start to then encapsulate the growth factor in specific domains within that polymer itself. So these different ways of manufacturing these things have a different influence on how these growth factors diffuse from the particles and scaffolds. So these soluble cues exhibit different diffusion rates from those particles and scaffolds, and and that's because they exhibit different degradation rates of the delivery vehicle. So you can enable this by putting them either in these small domains or directly in the polymer, the varied rates of the delivery of those growth factors. So just showing on the right-hand side is the cumulative release. This is for the VEGF, which is encapsulated directly in the polymer. Is pretty consistent. It's not linear, but it's pretty consistent over time. Where you can see with the cumulative release for the PDGF on this plot D, you can see that there is an initial release, and then it slows down, and then there is a second release. So the way that we encapsulate those growth factors into these different scaffolds can influence the way that they diffuse out. So there are different ways that we can present these. We can supply these cues inside of our polymers. So the first example here is non-covalent. So that's where we have this slow release of different cues, which are attached to the cells in there, so they will release slowly over time. Another one is a use of these covalent growth factors. So they are tethered endothelial growth factors, and they enhance this. It's a stem cell. Let me just get the definition of this one for you, because I forgot it. It's not a mesenchymal stem cell there. It is. Oh, I haven't written this on my notes. Okay, so let me see if I can type this in for you. So I thought this was actually mesenchymal stem cell. It's blastoma. So we can covalently tether those growth factors into there and get them to release over time. And then we can also use something which is pretty cool called an RGD peptide. The RGD peptide, and I'm going to write that one in for you as well, is called I don't know. Oh, I know why it is. I was like, I don't know why that is RGD, but it's because that each one of those are amino acids which are represented by their requisite lettering. So the RGD peptide actually is a motif responsible for cell adhesion in extracellular matrices. So we can use that specifically to introduce cell binding and spreading of those things as well. So one of the things that we can do as well is we can get the breakdown of our polymer scaffolds using enzymes in order to, using enzymes secreted by the body. So during tissue regeneration, fibroblasts, which you're aware of now, produce enzymes, and they break down and remove existing extracellular matrices. The major study is these matrix metalloproteinases, MMPs. The MMPs work by degrading the extracellular matrix proteins and the cellular surface receptors. They release these apoptic ligands, and they inactivate cytokines, chemokines and cytokines, and they play an essential role in cell proliferation and migration. So they incorporate these peptide sequences into covalently when we want to release these growth factors that are susceptible to the matrix metalloproteinases breakdown. So that enables the degradation in response to new tissue regeneration. So when we're thinking about a scaffold remaining in the body for just about the right amount of time in order to allow for repair, the incorporation of these cross-linkers, which are degraded by these enzymes, is an ideal way to do it. So we can also have the integration of proteins. So we have the cell adhesion proteins. So we can have that RGD sequence in there. And the important thing is with the RGD sequence is that we can acrylate or methacrylate on both ends, and that means that we can enable cross-linking into the network. So we have that ability to trigger the breakdown of that scaffold. And then we can also have these enzymatically degradable enzymes in there, peptides in there. So this is an example of a short peptide there. They're degradable by those metalloproteinases, and you can incorporate them again by acrylating or methacrylating in order to provide cross-linking into a network. So it must have cross-linkable groups on both ends to promote degradation. You know this now, really, in order to be an effective cross-linker, it's got to have either multiple points of attachment on each end, or it's also got to have an attachment right in the center of the molecule itself. So this is just a basic example of how you can enzymatically degrade PEG hydrogels. So if you work through this, the fibroblasts invade the adhesive metalloproteinase sensitive hydrogel. You get the migration of spindle-like shaped fibroblasts in there, responsible for that creation of the ECM. The cell invasion distances increase with time, The cell invasion rate is dependent on the proteolytic activity of the incorporated peptides. And the migration rate is dependent on the adhesion ligand density. So obviously the ability to migrate depends on the density of that scaffold that we've got in there. And that leads to the next one, where the cross-linking density of the hydrogels is influenced by cell invasion and those things. So what we've got is a number of different bioactive cues that we can use to enzymatically break down these scaffolds that we put inside the body, in tune with tissue regeneration at the same time. So we can also use mechanochemical factors in there. So the mechanochemical are bonds of this physical or chemical nature that affects cell state. So the two examples that we got there is the surface chemistry regulating the mesenchymal stem cell differentiation. And we know this now, that when we change the different surface of these polymers or materials, we can have different fates on the generation or the adherence of these things. That also affects when we're talking about differentiation of stem cells as well. So you can see that the stem cell differs whether they've got polyethylene glycol, this methacrylic acid, and ethylene glycol, or this T-butyl on the outside of their scaffold. And then we also know that changing the scaffold density affects the stem cell differentiation too. So if we can modulate that different cross-linking density in these polymer scaffolds, we can change the way that stem cells differentiate. So stem cells are a particularly interesting area, obviously, for lots of reasons, but the ability to control the way that they differentiate is especially important. And that's why you see the two examples there are published in Nature and Science, because it's on the forefront of what people want to know in order to get the right cells for the right application. So one example is tissue engineered skin, or this dermagraft. So this is actually taken from a diabetic, the pictures from a diabetic foot ulcer. So what they do is during the manufacturing process, they seed their scaffold with human fibroblasts, which will help for the regeneration of that extracellular matrix. The human fibroblasts, they obviously proliferate and they fill these interstices of the scaffold. The human fibroblasts then secrete dermal collagen, matrix proteins, growth factors, and cytokines, everything in there to promote the regrowth of the tissue. And then this, the three-dimensional human dermal substitute contains these metabolically active living cells. That dermal matrix forms around the scaffold during that manufacturing process, and after the application to the diabetic foot ulcer, the scaffold is absorbed. So you can see actually that's actually amazing how the ulcer has almost disappeared over the course of what looks like only a couple of months, right? So by creating this scaffold and seeding it with the fibroblasts, we provide the area where the foot ulcer was with all the important ingredients in order to regenerate. So that's one particular cool aspect. We can also look at things such as neocart and carticell, and these are cartilaginous tissue implants there. So the cartilage tissue is derived from the patient's own cells, and the carticell are cartilage cells used for the repair of symptomatic cartilage defects of the femur. And so that's medial and lateral defects, and that are caused by acute trauma. And then the osteo, so the incorporation of things such as, so in vivo, they culture those autologous chondrocytes. And so they have long culture times that are expensive, and they have questionable efficacy into their things. So how can this improve? Well, it can be improved by incorporating it into these scaffolds as well. So that's the important thing, is that when we're looking at just when we're looking at kind of regenerating a whole tissue, and whether it's a cartilage or whether it's a skin tissue, we know that it can be a benefit now to incorporate those things into these scaffolds to aid that regrowth. I'll give you a second to write that down. So here's another example. This is called osteocell. So the osteocell retains the natural scaffold of bone, and it allows new bone to grow on and through. So they have decellularized allografts. The potential problems with those is that they can get rejected. So the antibodies of the transplant recipient reacts with the donor antigens on the cell surfaces within the donor organ. So they can often be rejected. So that decellularization is a process of isolating the extracellular matrix of a tissue from its inhabitants cells. So that leaves an extracellular matrix scaffold for the original tissue to grow. So we can have embed in those different signals. So when we want the bone formation to occur, we know that we need specific signals and growth factors in there to direct the process. So these signals act on the cells within the osteocell and help direct your body's own cells to stimulate bone formation. So what we're doing is producing this scaffold that has got all the necessary ingredients in there. And then the cells inside that will then contain these viable MSCs, which can differentiate into these bone forming cells, which are naturally present in the body and are essential for that bone formation and the healing of the wounded site. Another way, another different way of looking at these things is tissue engineered blood vessels and using things such as Omnivo. So this is cross-linked ovine collagen with a polymer mesh endoskeleton. So ovine is sheep, so this is a xenogenic transplant. The polymer mesh adds strength and durability, and it's resistant to aneurysm formation. The collagen itself, the collagen coating on it stabilizes this it's non-antigenic, and it remains structurally intact for years. These things are highly biocompatible, and they encourage rapid incorporation into the host tissue, revascularization of the wall, which we want, and they have resistance to infection in there as well. And then the wall is also impervious to tissue ingrowth and lumen, so there's long-term patency in those things too. So then we can also use this ECT implants as well for these tissue engineered retinas. So the ECT is this encapsulated cellular technology. So these ophthalmic diseases are most commonly associated with aging populations. The issue with the current treatments for these things is that they lack kind of effective delivery or of the therapeutic molecules. So this encapsulated cell technology allows for continued controlled and continuous long-term administration of drugs and biomolecules into the eye. So they have this semi-permeable exterior capsule and internal scaffold. Sorry, I think that keeps clicking on. They have this internal scaffold with encapsulated cells, and these cells can be used to express therapeutics such as antibodies and fusion proteins and growth factors. So the good thing about using that semi-permeable capsule is that we can change the way that nutrient and waste is transported through the capsule, and we can monitor that therapeutic diffusion out of the capsule, and we can also isolate the cells from the host by sealing them within the device, right? So there is no biocompatibility or rejection issues there because it's not actually touching the host. And then this is, I believe, is the final slide. Yay. So this is an example of how they use these tissue engineered polymers for use in cardiovascular uses. So this is an example here is cardio wrap. So the cardio wrap is this bioresorbable sheet for post-surgical healing after cardiothoracic surgery, so a really invasive surgery there. And the material is made of this polyol lactide, or PLDA, basically. And it's placed over the pericardial defect for use as pericardium replacement in patients. So anyone should know that, I think you should know that the pericardium is that kind of membrane that encloses the heart. Using these things, they could use these things for kind of up to 6 months, especially for a replacement in patients where they require that reoperation. Using these sheets, they exhibit minimal inflammation. They're lightweight and they're transparent, so that allows you to visualize the anatomy of what's going on. They can be easily repositioned in wet environments. Obviously, you can adapt them, so you can trim them down, you can suture them. They remain impermeable up to 8 weeks. And then the sheet itself contains significant tensile strength, greater than 100% for the initial 8 weeks. And then that gets a controlled decrease in that strength over the course of 24 weeks, which is a 6-month period, right? So that is it. That is officially it. We have managed to get through 50 modules, so we can celebrate a little bit. So this is a plan moving forward from here on in. So I'm going to help you as much as possible. I'm going to give you, I think you have one more in-class homework, which I'm going to actually release to you today. So you can have a look before the in-class, so you get plenty of time to prepare. I'm going to actually help you with that homework. So I'm going to allow you to separate out into your individual groups for 20, 30 minutes or so. Then we're going to come back into the main room. We're going to have a discussion about how you've tackled it, what the answers possibly could be. I'm going to pretty much go through the answer. So I'm helping you. I'm doing your in-class homework with you. But I want you to really get a sense of how to answer those things, because you'll have a similar question in the homework. So hopefully you understand how to do that. Now, what's also really important is that I need, obviously, feedback for this course is particularly important. So we'll do the feedback on Wednesday. Anyone who's not here for that, I'll also post the link to the feedback, or at least give you the direction on how to access the feedback. So I'll give you time in class on Wednesday also to go through that as well. But it's really important because obviously this year is different to every other year, I think. We've had a lot of twists and turns in the course and a lot of things that we have to deal with, but I'm actually happy that we seem to have made up quite a considerable amount of time with minimal amount of stress, hopefully. You know, any kind of feedback is really valued through that. So I'll give you the instructions on Wednesday as well, how to do that. And then I'll also give you an insight. So the exam will cover everything from kind of the shape memory polymers in lecture 9 all the way up to where we are in lecture 15. As I did for the first exam, I'll also do a review of the things that are essential for you to know and things that are maybe not so essential for you to know, and we can go through that. Oh, and I should also say, for the feedback, I've done this in the classes before where if we get over 95% of people feedback, then I will put a bonus question on the exam, which is out with the exam marks, which will give you an extra % or 2 to your overall grade. Right? So the idea is there is some incentive, right, to give some kind of feedback on those things, but you have to kind of work for it in the sense. But anyway, we'll get around to that. So we'll set that up. And then, yeah, then we will do one more in-class homework. And then I will be around from now until that exam period If you have particular questions, please come to the class and ask the questions. You know, and I can help you through those things. Please use my office hours. Please message me. I try to keep up with those as often as possible. It's easier for me to do it through Canvas because I see them straight away. So I try and keep on top of that. So we've basically got a good few weeks now where you can get all your notes in order for the exam period as well. Okay, so that's it from me today. Is there any questions or anything I've not made clear?
Professor Mappet, I had a question about the homework.
Yeah.
So for question 8, it asked for like 2 changes to the PEO and PPO blocks that decreased to LST. And so I was looking at the lecture notes, and it talks about the PPO, but it doesn't really talk about PEO. And so I wanted to know if like you could elaborate on that, or
I think the general thing is that if there's any changes that you can exhibit for the pleroxamers in general. Okay, so I've just done it as these block copolymers, but you could also exhibit the opposite changes for the PEO as well, and that would have a change on the LCST, but they're not explicitly written down, if that's what you mean.
And then I had a question about like the increased concentration in water. I was kind of like confused about that.
Increased concentration in the water. What's that?
It's like the last bullet point on lecture 10, slide 31. It's like it's another way to decrease the LST.
Oh, right. Okay. Are you free during my office hours? I can probably go through that in a little bit more detail then.
Okay. I'll just come through during office hours.
Yeah, that would be perfect then, because I can sit down and actually look at the slide and go through that with you.
Okay, thank you.
Okay. Everyone else good? Yeah, it's Monday. Okay, thank you. I'll catch you on Wednesday.